One second. So if you look at the slide, the heading says is the fate of the drug after administration. So you already learned the route of administration in other years, maybe first, second, or third year, whichever year you learn. You know there are various types of route of administration which we actually use for administering the drug to the body. That depends on the type of the dosage form. So this diagram just gives us a very brief idea about, like overall idea about what happens to the drug, fate of the drug when. One second. So this picture just gives us idea what happens to the drug after administering to the body. It's a schematic representation. Okay. So just look at that. If at all it is an oral administered drug, so just let's imagine the drug is now administered to the body. So once it is administered to the body, oral GI in the GI fluid, what happens? The drug get disintegrated. That is tablet means it get disintegrated, dissolution followed by the absorption. Once absorption is done, it enters into a compartment called as the systemic circulation, and that systemic circulation is generally called as the central compartment. This you'll understand when I start with the topic pharmacokinetics later on, quite 2-3 chapters later we'll be starting that. So this central compartment, and it's from there, that's what I told you, the blood acts as a transporter. That means it carries the drug to the each and every corner of the body. So it reaches the central compartment, and in the central compartment, many things can happen. It can bind to the protein, that is your albumin protein, which are present in the blood. There are varieties, glycoproteins are also present. So there's something happens called as protein binding. So it can bind to the protein, okay, and there are also chances that drug may not have bonded to the protein, and then it is called as the free drug, which is appearing in the central compartment. If the free drug is there, this free drug has easy access to the other parts of the body because it's not bigger in size. But when it bonds to the protein, its size increases. So because of its increase in size, what happens? It cannot easily cross the capillary barrier. The central compartment is blood. Nothing but the blood is flowing inside the capillary. So every capillary has a capillary membrane, which the drug need to cross. Though it is permeable to most of the molecules, capillary membrane, because of the increase in the size of the drug after binding to the protein, protein bonded drug cannot easily cross the membrane. But the free drug has an easy access to other part of the body. So it can go to the different parts of the body where it can go and bind to the tissue reservoirs where actually the therapeutic action is not there, but it has more affinity towards the tissue. As you can see in this aroma, therapeutic action may not be there, but still since it gets distributed throughout the body, what happens? The drug can just go and bind to its opposite partner whom it loves lot. Okay, so it can bound to that proteins or macromolecules present in the tissue and just sitting in that part of the tissue. The other one is where it actually goes and binds to the receptors, and that receptor is exhibiting the therapy. I mean, after binding to the receptor, it exhibits the therapeutic action. So the therapeutic action happens only when it binds to the desired receptor. Okay. And also there's something called as unwanted side effect can happen. It can bind to the other tissues also where the same binding. Here also it binds to the binds to the tissue macromolecules. Here also it binds to the tissue macromolecules, but here it is causing unwanted effect. That is the side effect. So a free drug which is present in the central compartment can cross the capillary membrane and reach the different parts of the body and can start exhibiting the different action, which may be desirable or which may not be desirable or which may not have any kind of effect at all. So as in case of the center box, whichever I am showing. Finally, okay, after this what happens? It's not a sequence. Again, I'm telling you, once the absorption has happened from the after the administration process, many things goes on parallelly. As you can see the arrow marks in this diagram, multiple tasks are happening to the drug. Means it can happen, the therapeutic action can exhibit, it can cause unwanted effect, it can reach other tissue part where no effect is exerted by the drug. At the same time, if you look at this, that arrow mark which is coming out, that is output arrow mark. That means the drug is getting eliminated out of the body. That is a body in the sense, right now let us imagine the central compartment. So it is like clearance called as excretion. The first term clearance you may not understand, but it's another pharmacokinetic term which they use for the excretion process. At the same time, as even I told you, elimination is the combination of metabolism and the last class we discussed about the two major terms. One is elimination, other one is disposition. Elimination is combination of the that is metabolism and the excretion, and disposition is combination of distribution plus elimination. So that you just try to correlate here. So the free drug can enter into biotransformation process if it is it can undergo hepatic first pass metabolism, or it can go directly to the kidney for the excretion process. So anything can happen. That entirely depends on the nature of the drug. Fine. So this is what happens to the drug once it enters into the systemic circulation. So that's why it is called as the fate of the drug. The ADME, whatever we would have understood by this time, it is nothing but that is the fate of the drug which happens inside the body. And this which we cannot easily stop unless until we modify something with the original drug molecule. If you can modify the drug, may not be always chemically, but some way if you can modify the properties of the drug, then you can avoid all this happening. You can minimize all this unwanted. If you feel it is an unwanted effect which is happening, I can try to modify either by, for example, I don't want metabolism, the drug to undergo metabolism process. So, but we know that if it is sensitive for the hepatic first pass metabolism, it's not very easy to avoid it unless until we convert the drug into prodrug. Means chemical I need to modify such that it may avoid the first pass metabolism, and but it has to come back to its normal original form before it exhibits the therapeutic action. So it's not an easy task. For every drug molecule cannot be converted into prodrug molecules. So best way is I avoid the oral route itself. So I have the alternative route of administration. I can go with the this one rectal or transdermal. Okay, so like this I look into the other, I venture into the other options where I can avoid the biotransformation process. That means avoiding the first pass metabolism. Fine. So like this designing of the dosage form and based on the route of administration, we can avoid many of the situations like consequences which is happening to the particular drug molecule during the journey of the molecule inside the body. So then what is absorption? So that was just a brief on what happens to the drug molecule once enter into the body. So once the drug is administered into the body, so this one, for systemic drug absorption, drug must first absorb into systemic circulation by crossing the cellular membrane and then diffuse or transported to the site of action to exhibit the therapeutic action. Same thing, whatever I just showed schematically in the previous slide, that's been written in the sentence. So for any therapeutic action to be exhibited by the drug molecule, the drug should reach the systemic circulation by absorption process. Again, see, this depends on the route of administration. And if it, since the absorption chapter we are talking, oral route is the major route of administration, and in that oral route, absorption is the first step. So the absorption default comes in the oral administration. So for a drug to reach the systemic circulation, So absorption of the drug should happen to the systemic circulation first, and then from the systemic circulation to the different part. It is transported to the site of action to exhibit the therapeutic action. Let's understand its definition. Then what is the definition of the absorption? So before that, remember one point. The correlation exists between the plasma concentration of the drug and the therapeutic response. See, why we are so much worried about the drug reaching systemic circulation, because that is the drug actually which is trying to reach the therapeutic site. So there is a relation between the plasma concentration of the drug and the therapeutic response. There's definitely, that's why we use other term called as bioavailability, the rate and extent of drug reaching the systemic circulation. So that bioavailability gives us an idea about the therapeutic response of the drug. If the drug can successfully reach the systemic circulation, definitely we can expect it respective therapeutic response. If the drug doesn't reach the systemic circulation, we cannot expect the desired therapeutic action. That's why, in conclusion, there is a correlation between, or there is a relation between the plasma concentration of the drug and the therapeutic response of the drug. So absorption can be defined based on this, whatever the general information we gathered now. Is a process of movement of unchanged drug. Underlined, have underlined means there's some important thing about this word unchanged. From the site of administration to the site of measurement. Unchanged. The drug, when you administer to the body, there is a lot of chances that it can undergo changes. The changes can happen in the GIT also. The pH, whichever present in the pH condition of the stomach, for example, acidic condition, can change the drug molecule. It might degrade the drug molecule, or the enzymes which are present in the gastric environment can change the drug molecule. That should not happen. If at all the drug, unchanged drug, moves from the site of administration to the site of measurement, then that is measurement is what? Is a plasma. Blood sample is a very important sample. Generally, we use it for our biopharmaceutical and pharmacokinetic studies. So the unchanged drug, if it reaching the systemic circulation or the site of administration, the movement is called as the absorption process. Okay, so I repeat, what is absorption? It is nothing but the process of movement of unchanged drug from the site of administration to the site of measurement. Vinny?
Yes, ma'am.
Fine. Any doubt?
No, ma'am.
Manasa?
Yes, ma'am.
Any doubt?
No, ma'am.
Okay. So everybody understood no? What is the definition of absorption? I just want at least answer yes, ma'am. So it becomes even comfortable for me to go ahead.
Yes, ma'am.
Who is that?
Yes, ma'am. Maybe.
Okay. Siddharth? Can you define what is absorption?
Ma'am, absorption is the movement of the unchanged drug from the administration site to the plasma.
No. Repeat again.
Absorption is the process of movement of unchanged drug from the site of administration
From the site of administration
to the plasma.
No. From the site of administration to
the plasma.
Wait, Lipika. Lipika.
Yes, ma'am.
Can you define?
Ma'am, the process of movement of the unchanged drug to the site of administration to the site of measurement. That is.
It is, say it's a movement of unchanged drug from the site of administration. Okay, site of administration can be anything. Right now we are just imagining oral route. So that is the site of administration. To the site of measurement. What is usually the measurement, the area, which part of a body we try to usually sample is the blood. So you can also define the process of movement of unchanged drug from the site of administration to the plasma or the blood circulation is also called as the absorption process. So before proceeding ahead, you know AUC, area under the curve. Everybody have idea about this AUC, area under the curve?
Yes, ma'am.
Okay, other than Lipika, Sakshi,
Yes, ma'am.
Sakshi,
Yes, ma'am.
okay. Navya,
Yes, ma'am.
fine. So all are attentive. Just cross checking whether you guys are attentive or not. So this AUC gives lot of information. See, when we tell that absorption is movement of unchanged drug from the site of administration to the site of measurement. And we know the site of measurement is now is the blood. So what is that we use for assessing the drug in the blood is the plasma. We are going to separate the plasma from the blood, and in that plasma we are going to measure the drug present in the plasma. So we are using some analytical method like HPLC or UV spectrometer or colorimetry, whatever it may be. It depends on the individual drug molecule analytical part. So once you measure that plasma concentration of the drug, and I need to represent that data in the graphical manner. So the best graphical representation in the biopharmaceutical pharmacokinetics study is the plasma concentration time data. So for every time interval, we need to measure the plasma concentration. Give the tablet to the person, volunteer, it may be animal model or a human model, and start collecting the blood sample at different time interval, assess the drug in the plasma, and now that data, whatever you collect, that is the time and the plasma concentration, you're plotting it on the graph. So this is how the graph looks, and that the graphical representation depends on the route of administration. Every plasma concentration time curve may not look like a parabola like how you are seeing on the screen. This is a parabola graph for an oral administration. Okay, because in oral administration what happens? Absorption happens and then followed by the elimination process. So this graph may not be same for the IV administration. If the drug is administered by the IV route, okay, in that case, we don't get to see the absorption happening. The reason simple is in IV route administration, there is no absorption step involved at all because all the drug is administered at once into the blood circulation. So this line, the increased line which is not visible in case of the IV administration. The line just starts from the y-axis because all the drug at the 0 time interval goes into the systemic circulation. So lines start declining because of the elimination process. So the plasma concentration time curve is not same for all route of administration. Is this way? I want you guys to remember. This depends on the route of administration, but AUC is nothing but the area under the curve. Whichever the route you administer the drug, when you measure the plasma concentration of the drug with respect to the time interval, we are going to get definitely something called as area under the curve when you plot it on the graph. So area under the curve is nothing but the relationship between it gives the interrelationship between the plasma concentration of the drug in the plasma and the different time intervals. So this AUC gives lot many information with respect to our understanding. It gives pharmacokinetic information. It also gives pharmacodynamic information or the pharmacological information, or you call it as a pharmacodynamic information. So various parameters which you can measure from the AUC, as you can see in the slide. So this is called as a Tmax. We know that the time taken. Somewhere in future also we'll be talking in detail about this. And this is called as the Cmax. Okay, and you would have also learned something called as MEC, minimum effective concentration. and the minimum toxic concentration. And these are the pharmacological parameters, pharmacodynamic parameters. Tmax and the Cmax, these are all the pharmacokinetic parameters, something to do with the time. Okay, all that are called as the kinetic parameters. Even the T half, which we calculate from the straight line of the elimination phase, this is called elimination. We calculate the parameter called as T half based on the slope value, and that is also a kinetic parameter. So like this plasma concentration time curve gives us many information, from kinetic information as well as the pharmacodynamic informations. Okay, so I just wanted to show you this graph so that keep in mind, after oral absorption, depending on the route of administration, if at all absorption happens, then the plasma concentration time curve will be definitely somewhat like this, increase followed by the decrease. So if you can just look at the graph other part also, this line is very important for us, MEC. For any drug to exhibit its therapeutic action, the concentration of the drug in the plasma should be above this. If the graph is below this effective concentration, then what happens? It's not the drug, though the drug has entered into the body, you cannot expect the that is pharmacological action. So you want the dose should be given. That's what I told. No, what is the advantage of calculating the pharmacokinetic parameters? That helps in deciding the dose of the drug. So we have deciding the dose of the drug with all this basic information. So I decide the dose such that when I given the dose to the patient, that dose will cross the minimum effective concentration and definitely going to exhibit the therapeutic action. Again, the dose should be calculated such that it should not be excess. Excess in the meaning, it should not cross the minimum toxic concentration. We know what happens. To cross the minimum toxic concentration means it will start exhibiting the unwanted side, the toxic effect, which is expected to be happening above that concentration. And also, if you look at this arrow mark, gap between the MTC, sorry, arrow mark I am not drawn here, MTC and the MEC, the gap is called as therapeutic window. The region between the minimum effective concentration and the minimum toxic concentration is called as the therapeutic window. So within this only you expect the Cmax to be present. If the Cmax is below that, no therapeutic action. If the Cmax is above the therapeutic window, it results in the toxic activity. There's a one more arrow mark, as you can see, intensity are written. That gives the what you call the intensity of the effect. How much effect the drug is giving, the therapeutic action is giving. That is understood by the intensity or the peak of the graph. If the peak is very sharp, means the action is very intense. Okay, immediately you will get to see that action happening. If the let us imagine the curve is going very broadly and it declines slowly. The broad peak, that means intensity of action is very, very slow. Fine. So the intensity of the action can also be decided based on the sharpness of the curve. There's one more thing here which you can also understand is the duration. How long the action is exhibited by the drug. That is the starting and the end point of the action. So this will give us the duration of the action. From when it is start, there is nothing but the line where it starts, first time it touches the MEC, and the line where it ends, well, before it ends, no, where it touches the same MEC point, the gap between that is called as the duration of action. So how long the action is exhibited by the drug molecule is given by the duration of this particular gap, the region. Okay. So let's proceed. We started with absorption. We know now absorption is nothing but movement of unchanged drug from the site of administration to the site of measurement. That is nothing but the plasma. Okay. So once if you measure that plasma concentration with respect to the time, we can plot a graph called as the area under the curve. And this area under the curve gives us both the kinetic information as well as the pharmacodynamic informations. Okay, let's enter into the mechanism. That is, mechanism of drug absorption is the another. By chance, if the Zoom links, the 40 minutes get exhausted, no, you enter back with the same link. We are going to understand now the mechanism of the drug absorption. Before that, we know that inside the GIT tract, see, each and every part of the body is made up of cells. All that you are a better person would have learned in detail in the HAP. Okay, so this is the epithelial cell which are present in our GIT tract. That is nothing but GI barrier, the tract which runs from the esophagus till the anus. Okay, that completely is made up of the epithelial cell, and its arrangement are different from one region to another region. We know in intestine there are microvilli which is not present in the stomach. So like this, this anatomy and physiology of the GIT will vary from one region to the another region. That is a secondary aspect. But generally, there are two types of drug transport. So word transport is nothing but here is absorption. Okay, so no need to get confused. So we are talking about absorption only. It's nothing but the movement. The other word we can use is the transport of the drug from one region to the another region. The region what we are talking right now with respect to oral route is one is the GI fluid, and it is getting, the drug is moving from the GI fluid by crossing the barrier, that is the epithelial barrier present in the GIT, and then it enters into the systemic circulation. This movement can happen either by the two major mechanisms. I can call it as transcellular and the paracellular. Transcellular is nothing but through the cell. The diagram is self-explanatory, as you can see in the diagram. Through the cell, if the drug molecule try to move through the cell, then it is called as the transcellular transport. The other one is the paracellular, the movement of the drug molecule between the gaps. That's what I told you, the epithelial cells of the GIT tracts, the arrangement are different from one region to another region. So depending on that arrangement, there is this possibility of this kind of gap generated between the cell, but it is one of the process which happens in minimum, very minimum compared to the transcellular. So transcellular is the major mechanism by which the drug enters through the epithelial cell. Paracellular is for very few exceptionals because that depends on the size of the drug molecule. So between the gap is called as paracellular transport. Through the cell is called as the transcellular transport. So we are entering into the mechanism of drug absorption. Generally, it can be broadly we can say it is by the transcellular transport mechanism or by the paracellular transport. So transcellular through the cell, paracellular between the cell. So this, before understanding the in detail the mechanism, there is some theory behind this movement of the drug. Initially, when they started carrying out the experiments to understand how actually the drug absorption goes on, the different scientists come out with the different theories. So two theories are well accepted now. The one is called as lipid bilayer or a unit membrane theory. The other one is called as the fluid mosaic model theory. So the two theories based on which we try to justify the movement of the drug from the site of administration to systemic circulation in the GIT is either by the theories are lipid bilayer or unit membrane theory. The other one is the fluid mosaic model theory. So let's briefly understand. We are not going in detail. Look at the lipid bilayer theory. Everybody knows which lipid bilayer they are talking about. Phospholipid bilayer. We know every cell is made up of cytoplasmic membrane, and this cytoplasmic membrane is nothing but the phospholipid bilayer structure. We know that. So when I say epithelial cell in the GIT, whatever this epithelial cell I'm telling, definitely it's a cell. So cell will have its own cytoplasmic membrane, that means phospholipid bilayer structure. So when the drug has to move from this region to another region, okay, it has to cross through the phospholipid bilayer structure. So they are talking about the lipid bilayer structure here. So this theory considers what? We are talking about the first theory, is also called as a unit membrane theory. That is the lipid bilayer theory, also called as the unit membrane theory, mainly considers the plasma membrane structure, which is composed of phospholipid bilayer structure. And we know that this phospholipid bilayer structure is having a polar group and the non-polar group. That is a head and tail. Head is polar and tail is the non-polar. So this head groups of the phospholipid facing the protein layers and the hydrophilic tail groups of the phospholipids, which has alignment is such that that head is towards the upper side So I think the diagram is here. So as you can see in this diagram, all the heads are arranged on the upper side and the tails are inside. So this is hydrophilic structure and this is the hydrophobic structure. Okay, so unit membrane theory mainly considers the structure. Which structure? Phospholipid bilayer structure. So whatever knowledge you have gained about the cell structure, you can start using that knowledge here. So this structure, according to this structure, it tells what? The diffusion of the water or the smaller weight such as urea and the certain charged ions will happen through this structure. Water structure does not account, means it will not support this layer, that is bilayer theory does not support for the diffusion of water, small molecular weight, that is urea, and certain means the hydrophilic movement of the hydrophilic molecules through this structure is not substantiated by theory. That means what? The other one. This theory tells that only those drug molecules or the molecules which are lipophilic in nature can there's a lot of disturbance. I'm taking a light difference. Yeah. So according to this theory, it justifies only the movement of the hydrophobic molecule because phospholipid bilayer structure is hydrophobic in nature. It has both hydrophilic and lipophilic in nature, but the drug molecules or the molecules which are more lipophilic in nature, they can easily cross the membrane because of the phospholipid bilayer structure. So it doesn't give any accountability for the diffusion of water soluble molecules like water itself or smaller weight molecular weight such as urea and the charged ions. So it mainly justifies the movement of the hydrophobic molecules. So this phospholipid bilayer structure is, we are not going in detail actually. We know it has integrated proteins, it's a bilayer structure. It is having the phospholipids, cholesterol, glycolipids. It has the proteins or integral membrane proteins, all this. Okay, each and every structure or parts of this membrane definitely plays important role in the movement of the drug molecule while during the absorption process. It just as an additional information, I have just taken this slide for your just to recollect whatever you would have learned. Same thing you can apply here. The second one is the fluid mosaic model, which actually talks about the fluidity of the membrane, means the flexibility of the membrane. So this is mainly it is based on the transcellular diffusion mechanism. So according to this model, so according to this model, the cell membrane consists of the globular proteins. Remember this structure, I told you there are many types of integral proteins. These are the integral proteins which are buried inside, which are outside, they are called as peripheral proteins. These are called the peripheral proteins. So these proteins which are present in the membrane plays different roles. Okay, so according to this fluid mosaic model, what it says is the movement of the drug molecule, that is by the transcellular, okay, for the polar molecules, especially the polar. The previous one, I'm disturbing the slide again. The previous lipid bilayer theory mainly talks about the movement of the drug molecule that is transcellular only, but based on the lipophilicity. And that lipophilicity is correlated with the phospholipid bilayer structure. That's why it is called as lipid bilayer or unit membrane theory. But in case of fluid mosaic model, they are mainly